any of the handouts that I've got up here that you got last week. You got 5 handouts. You got a blue one, a green one, a purple one, a manila one, and a bright pink cherry colored one. Everyone got those? Okay, because we'll talk about most of them except for the blue one, because that was what you talked about in the library with SciFinder. So the first thing I'm going to go over of those handouts is your syllabus, which is the manila one, okay? So a couple things there. I'm going to kind of have you guys go ahead and read through them, but I'm going to hit the highlights. So for the independent project, we're going to have 4 sections of them. My Tuesday morning, Gilmore is Tuesday afternoon, my Thursday morning, and Johnson Thursday afternoon. Okay, so you should be in one of those sections. You should be here for lab lecture. First deadline for you is we're going to talk about what's going to go into your proposal, but if you're in a Tuesday lab section, your proposal will be due on March 10th. If you're in a Thursday lab section, your proposal is going to be due on March 12th. Okay? And so we have to have your completed proposal with all its pieces by those dates because we need them before spring break because we prep everything over spring break. So while you're on spring break, feel for me, I'm not on spring break. I'm training. Okay? Just my little sympathy plea. So basic of the calendar and points grading wise for the project. So last week we did our library training. This week in lab you're going to be getting your proposals all together. First you got to get your literature together, then get all the pieces of the proposal together that I'm going to go through today. Get all that set. Then next week you're going to be submitting your project proposals before spring break. Then right when you guys get back from spring break, straight away we'll start working on your projects for 5 weeks, and then your final report that you're going to write is due tentatively the 24th of April, the last Friday of classes, okay? As far as grading for the projects, there's 3 grades. There's the proposal that you write is 50 points. The project report and the lab work you do is 150 points, and then safety and technique is 50 points. So there's 350 points possible. Sorry, actually, there should be 250 points possible, not 300. 250 points possible for the project, okay, from those 3 grades. And I'll let you read the rest of the syllabus, but basically you've taken Chem 255, 256A, be safe in lab, follow instructions that you've been given as far as keeping a notebook, all that kind of information is in there, okay? All right, so now the part of what I'm going to cover today is all going to be in this cherry packet because this is kind of the basic rules, regs, all the instructions for the project, all right? So, as we go through here, first part talks about basic premise of the project. You want to propose an organic synthesis, since we're in organic lab, so it needs to be related to organic chemistry. Typical length of synthesis is about 3 steps is what you want to propose, and you usually in that proposal want some sort of carbon-carbon bond forming reaction that is going to take place in that proposal, okay? So we propose the 3 steps. It's not, when you actually go and carry out the lab work, it's not a race to see who can get through the 3 steps first. What we want is quality, not necessarily quantity. So if you can complete 2 steps and do a really good job with those 2 steps and do really good with your characterization and make sure everything is really complete with those 2 steps, that's better than doing 3 steps really fast and having no idea at the end what really happened, okay? So keep in mind as you go along, quality is most important. The other thing that we want you to learn in doing the project is independence in the lab, and you being the driver now for the project. So instead of us having a syllabus that says this week you're going to do this experiment, this week you're going to do this experiment, this week you're going to do this experiment, those 5 weeks that you're working on your project, you're the one driving the experiment, so you need to think about each week for lab, what do you need to have prepared, analyze the data that you collect the week before so that you are ready to go on your lab day with the next step or whatever needs to happen. If you need to do a column to purify things, or you need to run the first step over again, all those things you guys have to be in charge of and be the drivers for, okay? So just make sure that you're always on top of your project and keeping things moving along for your project, depending on what's happening with it. And after the first week, once everybody starts, you're all gonna end up in different spots at that point, because once everyone starts something completely different, where you end will be completely different as well, okay? So I've already talked about this. March 10th, Tuesday labs, proposal due. March 12th, Thursday labs, proposal due, okay? So make sure that that is handed in before you leave for spring break. And I'm gonna give you a couple, at the end I'll give you a couple even more specific deadlines for some of the pieces. In a little bit here, so under section 2 here, it also talks about a chemical order form where you're going to put together all the reagents that you need for your proposal. That needs to also be emailed to me before you leave for spring break, because that is the meat of how I'm going to prepare all the chemicals for you for your project. So make sure you get that to me, okay? And again, I'll give you at the end some deadlines for that. For your for each step of your project then, so we kind of have set you up for the project in what we did with Experiment 20. So each step that you do with your independent project, you're going to need a pre-lab written ahead of time, and you'll need your TA or your instructor's signature on that pre-lab. So not just the TA, because TAs may or may not know exactly what you're doing, but the instructor needs to sign off on your pre-lab before you start that step. And it's really up to you now, because we're gonna have people in all different directions. It's up to you to go to your instructor and get your pre-lab signed off before you start a new step, okay? So each part you're going to need an experiment or a pre-lab. You're also going to need experimental for each step, and then you'll need your mini conclusion summarizing what happened in that step, okay? And so in that mini conclusion, even if we're repeating this first step over again, still summarize what happened in that mini conclusion, that's going to lead you to rerunning that step, okay? So you don't want to wait till, you've run that step and then deviated off onto another step and then blah, 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 blah before you write that mini conclusion. Once you've made a decision point, you want to write a mini conclusion, and that needs to summarize that decision point, all right? And then at the end, we will write a final conclusion, okay? And so that final conclusion is going to discuss what was done, what wasn't done, and why things happened. And then you're also going to turn in your notebook with your pre-lab experimental mini conclusion. You'll turn in all your spectra and data that you've collected, and then you will turn in that conclusion as well. So so you thought your experiment 20, your notebook was fat from experiment 20. It'll be equally as fat or larger from the independent project, okay? So I've kind of already discussed all these. So 4 is make sure you have a complete experimental in your lab notebook for each step. The other part with our 3-step synthesis is then you want to have, you don't want to just run each step without characterization. So you want each step to then be fully characterized at the end of that step. So use IR, NMR, probably GC mass spec as well as whatever other characterization is mentioned in your precedent or that your lab prof or whoever you're doing the project for thinks you need to do, okay? So at the end of each step, make sure you're getting this full characterization. Don't wait till the end to do it. You want to do it at the end of each step to make sure you're ready to go on to the next step. And then number 6 is talking about this final summary that you're going to write. And when we get closer to that, we'll talk about that, okay? Unless you are told otherwise, you're generally going to start with 10 grams of material for your first step, okay? But double check with who you're doing this project for, whether you're lab prof or you're in, you know, my section but doing a Johnson project. Find out exactly how much you're supposed to start with of your starting materials, okay? Be very clear about that, because you can't go on and figure out the rest of the chemicals that you need unless you know exactly what your starting point is, okay? So make sure from the get-go, make sure this week you are very clear about how much starting material you're going to use, okay? So this is a guide if you're not told otherwise, but most of us are going to tell you exactly how much to start with, okay? And then the bottom here, we're going to talk about this green sheet here. Part of that green sheet is chemicals that we can't use because of the hazards and toxicities involved, or chemicals that really have to be used with care because of the hazards, toxicities involved. Okay, so I'll talk about that here in a little bit, but basically anything that is really hazardous or reaction conditions that are really hazardous, any reagent or reaction conditions we're not going to use. Okay. All right. And so I'm not going to read through that whole paragraph of things, because like I said, we're going to cover things here on this green sheet here in a second. But reiterating the points for the grading, it's 50 points for the proposal, 50 points for lab safety and technique, and then 150 points for the lab work and the final report and conclusion in your notebook that you hand in. So kind of all of that project conclusion, everything in your notebook, analysis of your spectra, how you've handled the project, all go into that 150 points, okay? So it's not just all written, it's everything with that project, how well you've independently driven your project. Now, in independently driving your project, what you don't want to do, though, is not check in with your lab prof. So you need to constantly keep your lab prof informed of what you're doing, and when you're working in the lab, your lab prof needs to know that you are working in the lab, and they're probably going to be in there helping you work in the lab, okay? So it's going to be a different in-lab situation and open lab situation than it's been before, because you guys are all doing things that are different, and they're typically, unless you are collecting a melting point or collecting an IR spectrum or prepping an NMR sample, you're going to need someone in the lab with you as you're doing that. So the point person from now on is going to be your lab prof. If they aren't available, then we'll coordinate that maybe I potentially am available to help out. But first and foremost, you want to make sure you're making contact with your lab prof, okay? And then on April 24th, last Friday of classes, again, is when your final report is due, as well as any samples that your prof or whoever you're doing the project for wants. All of that is all going to be collected by then. And the Thursday, so April 23rd, is the last day of anything in the lab. So you need to make sure that you are completely, and even before then, you're probably going to be done in the lab. You have to be checked out of the lab by April 23rd, okay? So usually at that point, that's when samples are going to be handed over, is when you're checking out a lab. So that Thursday before, everybody needs to be checked out, okay? Academic honesty, remember, you know, you want to do your own work. The proposal should be your own work. What you do in lab should be your own work. We shouldn't have two people doing the exact same thing for projects. Everybody should be doing something different. There is going to be similarities in what people are doing, but they shouldn't be doing exactly the same thing all the way through, okay? And then make sure that your work is really your own complete independent work. So as far as references for the proposal, you're going to propose these 3 steps, okay? You need literature precedent for each of those 3 steps. So you need something in the literature that tells you how to do those steps, because you don't have the how-to. It's not going to necessarily, unless you look out, going to be in your lab manual. So you need to find the experimental details for the how-to of doing those 3 steps. And so make sure in finding those literature precedents, you're finding experimental that gives you enough detail to actually do the steps. So as you're reading that experimental, make sure, like, think about, can I take this experimental and do something in the lab with it and understand what I'm doing, okay? The other thing, as you're finding these precedents and going through what each of these steps are, the whole process along the way, your lab prof needs to be approving what you're doing, okay? So don't wait till your proposal is completely written and ready to go, and you go to hand it in, and then you're showing your lab prof for the first time what reactions you're running and what your project is. From the get-go, your lab prof needs to approve what your project is, and then as you are proposing each of those 3 steps, they need to approve what those 3 steps are and approve of the literature precedent that you're using for each of those 3 steps, okay? So on the 4th page and 5th page of the pink sheets, there's lots of general references for organic reactions and procedures. I've gone and marked the ones that we showed you in the library last week. So the ones we showed you are 2 organic reactions, 3 Marsh's Advanced Organic Chemistry, Larocque's Comprehensive Organic Transformation, which is 4, Vogel's A Textbook of Practical Organic Chemistry, 10 Fieser reagents for organic syntheses, and then on the next page, number 16, which is the Aldrich catalog, which doesn't necessarily have to be a paper resource, which mainly most people are going to be using online, which is fine. And then the Curdy book, Strategic Applications of Named Reactions, okay? So those were the ones we showed you in the library. There's a lot of other references in there that you can use as well, depending on what you need in putting your project together. Here's some useful chemistry journals that, depending on the year, we have access through the library, which are good organic chemistry journals, okay? There may be others that also can be used in addition to this, okay? So just to give you an idea of kind of when you're finding literature, is that something that's going to be reasonable? Is it going to be a journal that can be used for finding a procedure, okay? And there, like I said, there are others beyond that list. So I'm going to go through the pieces of the proposal and then show you the checklist and then give you some deadlines, and then I'm going to go through some examples of all these pieces that I have showed you, okay? So for the proposal, make sure your name's on it, generally, good idea, date, lab section. State the goal of the project, so why are you completing this project? Is it to make something that is important for Dr. Gilmore's research or Dr. Johnson's research or Dr. Zanford's research? Is it to develop a project for curriculum development? What is the reason that you are doing this, okay? Then you want to make sure you give a reaction sequence for each step, showing the starting materials, reagents, solvents, and conditions, and give some summary to that reaction sequence too. So I'll show you that here in a little bit. And then you need to show the literature precedent for each of those steps. And if your literature precedent isn't exactly what the step is that you're going to do, then you want to make sure you show what was proposed, what was the literature precedent, and what step then you're proposing related to that literature precedent, okay? Then you're going to give a table of physical constants, so just like you've done before in your pre-labs, but you're going to give it for the entire 3 steps in your proposal. So you're going to write a big physical data table. Make sure you have boiling point, melting point, density, molecular weight, and then you're going to give amounts, and you're going to give moles of what you're proposing to use, and you want to include in there the physical properties for all your products too, as best as they are known, all right? Then you also are going to have a table. So number 5 says that you're going to have this one table with all these things in it. It's really hard to get everything into one table, so I recommend splitting it into 2 things. You'll have your regular physical data table that your regular table that you've always prepared. And then you also need to have a table where you list the toxicological properties and the hazards of the chemicals that are going to be used in the compounds that are going to be made, okay? So you're gonna have 2 really big, complete tables of all this data. And a really good reference for that, if it's a chemical that comes from Sigma Aldrich, use Sigma Aldrich to get that information. It has all the physical constants. It also has hazards and toxicological information on the website. And if you need help with that, let us know. We can show you that. Then you need a separate list of all of the amounts of starting materials and reagents and solvents that you are going to need for your proposed synthesis. Okay, and I'm going to show you what that list looks like. It's an Excel spreadsheet, and that's one of the things that I'm going to send you tonight. Okay, and then that's one piece that you need to send back to me before spring break. Okay, then you're going to need a list of any special equipment. So make sure anything besides what's found in your lab drawer, you list that. And then you need to give you need to attach photocopies of your literature precedents and the full precedent. So don't just give me the experimental section of that precedent, but you want all of the pieces of the precedent, okay? And then make sure you include in there the supplementary information as well, okay? And then finally, a PowerPoint slide that shows what you're going to be doing for your project. And I'll show you, I have a template for that, so I'm going to send that to you tonight, and I'll show you that here in a minute. When you go to finally hand in all of your proposal, you want to make sure you have 2 copies. So you're going to hand in 2 copies, okay? We're going to grade one of them and give them that one back to you, and we're going to hang on to one of them so we know always what you're doing in lab and what you're proposing to do. So make sure you hand in the copies. You make the copies. Don't give us one and expect us to make the copy. Okay, you hand in the 2 copies for them. Okay, and so on page 7, first of all, is an example of how you'll use literature precedents that aren't exactly the same. So, for example, you wanted to carry out this reaction, okay, this alkylation reaction. You found, and so you're going to do this on a 6-membered ring. You found a precedent that shows it for a 5-membered ring. So this would be a reasonable precedent to use, okay? So in your proposal, make sure you show the proposed reaction, and then make sure you show the reaction that was shown in the literature, and then give the proper citation. And you want to make sure that you're using a citation that is up to date with the ACS style guide. Okay, so make sure you use the style guide, which is available on the ACS database that Dave O'Brien showed us last week. Make sure you use that style guide for citing the references correctly. Okay, and in that reference, you don't usually give the title. So there's multiple ways of using the references. You don't usually give the title of the paper. Okay, so make sure you're using a citation that doesn't involve the title of the paper. Okay. So now here on the bottom is that list I just read to you on page 6. Here is basically a checklist for all of those things. And again, remember your chemical order form you're sending to me and your PowerPoint slide you're sending to me. Okay, so you're going to have printed copies of these. And then you're also going to email me the file for the order form, and you're going to email me the file for your PowerPoint slide, okay? So with that, I'm going to give you guys some due dates for that, okay? Because I need all this in time to get things prepared for spring break, okay? So you're going to email me chemical order form and your PowerPoint slide. And so I need this by 12 p.m. and hopefully sooner than that, but definitely by 12 p.m. Thursday the 12th. Okay, so the last Thursday of classes before spring break by 12 p.m. If you're in a Tuesday AM lab, the Tuesday, so Gilmore's section, so my morning section on Tuesday, Gilmore's afternoon section on Tuesday, or the Thursday AM lab. Thursday afternoon, we're talking about your lab time, okay? So I need that for Thursday afternoon. I need it by 3 p.m. on the 12th for the Thursday afternoon lab. Okay? And again, you're going to get an email from me tonight, but my email is smitht@hope.edu. Okay? Now, make sure when you go to send it to me that you are ready to send it to me. So I don't want, like, you send it to me and then you're like, oh, wait a minute, I need to make corrections. Here's a new one and here's a new one. Like, I want the final version because it's very hard to keep up with what is the final version for all of you guys, okay? So make sure when you're sending it to me, you have talked about your order form and your PowerPoint slide with your lab prof, and you've gotten everything approved and they've gone over everything to make sure it's all ready to send to me, okay? All right, so now what I'm going to do is I'm going to go through an example of a proposal, okay? So in this example, we've got our name, which is blackened out to, protect the innocent here. Lab section, date, okay? Then a purpose, a goal, a reason for doing this experiment. So this person was doing something for Dr. Johnson's lab, so they gave the reason why it was being proposed to be synthesized, okay? And then you're going to break down your proposal into each of your steps, okay? So step 1, give a paragraph describing that step, what is going to happen in that step, okay? And you don't need to give 15 mils of ethyl acetate will be used as the solvent, and we'll add 5 grams of ice and blah, 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 blah. We don't need every nitty-gritty experimental detail, but we do need the basic gist of what is going to happen in that step. So if the reaction is going to be carried out at minus 78 degrees, that would be good information for us to know in this information, okay? Giving a basic description of this step, a Wittig reaction is going to be carried out between this thing and this thing, and after the crude product is isolated, then a column will be run to purify the product, that kind of information, okay? So just give a basic paragraph summarizing the step without giving unnecessary experimental detail, but kind of out of the ordinary experimental detail would be helpful. So, cooling it very cold, heating it at reflux for 18 hours, using a par shaker at 30 psi. Those kind of things are important, okay? And then you want to make sure you Chemdraw, so we're going to show you how to use Chemdraw this week. Chemdraw appropriately and correctly what the reaction scheme looks like, and then make sure that you give the appropriate citation for it. If you're following the same procedure as what's in the literature precedent, then you can say identical reaction, but what the differences are going to be. So like in this case, they used benzene as the solvent. They proposed to use toluene because benzene's really not a great solvent to be using, okay? So if you here were proposing a similar step, then you would show the step you're proposing and then go and show the step that is proposed in the literature precedent. So you'd have two two Chemdraw things to show there, okay, versus just one. Yes?
And that was for if they're identical, or if they're...
So if they're identical, you don't need to show the reaction scheme again. But if it's not exactly what is in the literature precedent, then you want to show what you're proposing and then the literature precedent's reaction. Does everyone understand that part?
But that is adequate right there. Like, if your thing is just something minor like benzene.
Yeah, like they're
versus
using a
solvent.
different solvent, it would be fine to just put a little note there instead of redrawing the whole thing to highlight this one thing right there. But if it's, you know, like in the packet, the cherry packet, where it's, you know, a carbon difference, then yeah, you should show what the reactants and products, you know, show a different reaction scheme. Okay? So you're going to do that for each of the three steps that you're proposing. You're going to write a paragraph describing it, show what that step is that you're proposing, give the literature precedent for it, and make sure the citation is correct based on the ACS style guide. Okay? Okay, so here's the other steps that were proposed, the rest of step 2, and then finally step 3. And then it's okay with me, and you can check with your lab prof if it's not me, that at the end of the proposal, then you could just put a list there of other special equipment that you'll need besides what was in the lab drawer. Okay, so you could just go ahead, if you have room, just go ahead and put your list there. All right. So then you need your physical data table for all of the things you are using and the products you're making. All right, so even for those products, whatever you can find, put that information down, even if it's, you know, your proposed yield, okay? And then this is where I was saying it might be easier to actually then do a second table to put the hazards or toxicological properties, because it gets to be a pretty extensive table. So if you think you can fit it all in, try, but it may be easier to do this as one table and then cut and paste this column and make another table where you have all that. Yes?
Do we need the CAS number too?
Yes, the CAS number is a good idea.
Where did we get that again?
Aldrich.
Oh, okay.
If it's a known compound that's
Right.
registered, it'll have a CAS number.
Okay.
And you'll need that for your order form anyway, so that's why it's useful to have the CAS number on there. Okay? All right. So now one thing you need to take into account here. So looking at like this proposal, let's look at. You need to take into account the literature precedent, what it's telling you about yields. Okay. So in the first reaction, you propose this reaction and say you were going to start with 10 grams of your diethyl L tartrate, and that was your limiting reagent. Okay. Then you get an 89% yield. Well, first of all, everything in this reaction was based off of you using that 10 grams. Okay. So you want to make sure, keep your mole ratios. When you're looking at a literature precedent, you're probably going to have to either scale up or scale down based on what you're using. Make sure you keep your mole ratios of reagents in the right mole ratio. Okay? So you got to calculate the moles of everything you're using and make sure that what you're proposing and what you end up using when you do the reaction is the same mole ratios as what was used in the literature precedent. Okay? Now for solvents, you want to keep the same concentrations. So make sure that your reagents have the same concentrations as what you had in the literature precedent. Okay? Then the other thing is you've got to use these yields that are proposed to then figure out how much you're going to have to go on to the next step. Okay? So in this case, we wouldn't say that we're going to have 100% product to go on to the next step. We're going to only have an 89% yield. So figure out what that 89% yield is. That's how much starting material you have for step 2. Then go on and do your calculations for step 2 of what you're going to have and what you're going to use. Okay? Everybody see that? Okay, so mole ratios need to stay consistent for reagents. Concentrations need to stay consistent for the reagents in those solvents. Okay? All right. And that's in here, you're going to fill in that information based off of those calculations that you make, all right? Same with the chemical order form. So this is the chemical order form that I'm going to send you, except for it's going to be blank. So I'll show you that here in a second. You want to make sure you give the chemical name and spell out the whole name. I do not want formulas here. So I'm taking your list and I'm going to make one big huge list of everybody's chemicals all together. So what I don't want are formulas here. I need names, okay? The other thing I need is if your name starts with numbers, put those numbers at the end, okay? So this 2,2-dimethoxypropane, or 2,2'-dimethoxypropane. Instead, what you would type is dimethoxypropane, comma, 2,2' dash, okay? So move the numbers to the back. You don't want to put them in the front because then it makes it tricky for alphabetizing things, all right? And so for each of the chemicals that you're going to need, you're going to list the chemical name, the CAS number, the amount you need, the amount on hand, and I'm going to show you in a second how to get that, quantity to order, what you're going to order it from, and then the catalog number. Then you're going to give your name and your lab prof's name, and then what step it's going to be used for, okay? So if there's something like, we use this step in that, you know, if there's something expensive or maybe back ordered or something like that, and you're going to use it in your third step, we're probably not going to order it until we know for sure you're going to actually do that third step. So we don't want to order a super expensive chemical for a third step if you're not actually going to get to it, okay? So it is important to fill in where that step's going to come from, okay? So I'm going to show you right now some things that you're going to need for that order form. It's slowly coming to life here. All right, so here's our order form, okay? You'll fill in the information. Like I said, I'm going to send you, this is the file I'm going to send you, is this blank file. Now the other thing, keep it in Excel. Don't transfer to a Google Doc, okay? Because it makes it really hard to cut and paste things. I need it to stay in Excel. I need you to send me back Excel. Don't change formatting or anything like that, okay? I need it to stay the same because I need to be able to take your order form and cut and paste it into the master. So don't make any changes really to it unless like maybe you need to make a margin bigger to fit the name in or something like that, okay? But really don't change the formatting of it. I need it to stay just as it is, okay? So where we are going to get information for you being able to figure out what chemicals we have on hand or not is there are two things I'm going to send you, okay? We're in the middle of a transition of our inventory system in the chemistry department, okay? Not everything is in the inventory system. So I'm going to send you an Excel file, another Excel file that has the list of all the chemicals that are in our big lower stockroom, okay? So you're going to need to check this Excel, and that's what's up here. You're going to need to check that Excel file for the chemicals that you need, and you're also going to use this guy here. And so the information for getting to this website is over on the board. So you need msdsonline.com, and then the login is over there, chemordering@hope.edu, which is just a Hope email address, and the password is Hope, okay? So you also need to check this database. And so to check it, what you're going to do is type in whatever you need to look for. So we're going to look for hexane, and we need to find, we're going to say we're going to search for it, and then we're going to go down to this list and click on hexane, okay? And then what's really helpful is there's the CAS numbers in here for it. There's a lot of information in here for it, but it also tells you where you would find it, okay? And so down here in this inventory, it tells you the room of where you would find it. Now, one thing that I'm hoping that they're going to add to this, and I've asked to be added to it, but it hasn't been added yet, is also to tell you how much is there, okay? So you want to list the room of where to find it, and you want to list how much, okay? Now I'm going to give you kind of a hierarchy of rooms, okay? So we don't want to just put down any old room in the entire science center, because biology probably doesn't want us going and taking their chemicals, okay? So places we can get chemicals from. 3121, which is the organic stockroom. 3109 and 3129 are the organic labs, okay? Then if you're doing a project for Johnson, his lab is 3116. Gilmore is 3120. Dr. Ackerman is 3122, and Sanford is 3126, okay? 3123 is a possibility because our refrigerators and our freezers, our refrigerator and freezers in there. So that's a possibility. Then after that, write down 1033. So if something pops up that says 1033, that's the lower stockroom. So if it pops up here as 1033, then you want to check the Excel spreadsheet. Okay, double check. Okay, because they can pop up here, but the master list for what's really in there is that Excel spreadsheet. Okay. Then after all of these, then if you aren't finding it in one of these rooms, then check with your lab prof if you find it somewhere else in the chemistry part. Okay, so chemistry is second and third floor. Okay, so they're going to be either 21 numbers or 31 numbers. All right. But if you're going to do something here, check with your lab prof. Otherwise, if you can't find it in something that is chemistry, leave it blank. Okay. All right. So I'm going to send you the link to this website, but I've also just given you the website, and I'll send you the login and the username. Okay. Or the login and the password. All right, so that as far as checking our inventory, that's where things are going to come from. Then you want to then look for, start with Aldrich as far as finding supplier information. Now, on that order form, you want to put supplier information for all of the chemicals, whether you think it's in the building or not, because the inventory may be wrong. Okay, so still put that ordering information in there, because sometimes there's chemicals that are slightly related or different different specifications, and we want to make sure we're ordering what you need to have ordered. Okay, so make sure you put that ordering information in there. Okay, now I'm going to put a list up here of other possibilities. If you can't find it in Aldrich, these are other possibilities that possibly could be used. So Acros, Alpha Acar, Lancaster. So you'll want to write this down. GFS, Fluka, TCI, and there's lots of types of TCI. So there's organometallics and catalysts and all sorts of different branches of it. Faults in Bauer, so it's P-F-A-U-L-T-Z, and Bauer. MP, and then Matrix Scientific. So these are other possibilities, but really make sure that you can't find it in Aldrich first, or make sure that it would be like cost-wise better to order from one of these or something like that. Aldrich chemicals we can get pretty quickly and pretty easily for the most part, unless it has to be delivered by truck. A lot of these usually are more expensive because we won't be ordering a lot from them, so we have to pay more in shipping. And it sometimes takes longer to get them, okay? So Aldrich should be your primary, but here's some other possibilities. The other thing you want to keep in mind is $1 per gram, okay? So if what you're ordering is more than $1 per gram, check with your lab prof to make sure that's really a good idea, okay? Now if it's a catalyst, you know, you're not going to use a whole gram of a catalyst, you'll use 20 milligrams of it. So maybe that's okay if it's expensive. But if it's, you know, you're going to use 10 grams of it and it costs $25 per gram of it, it may be something that's going to be a little prohibitive. We're probably not going to pay $250 just for your starting material, okay? All right, so one other thing I want to show you is then the PowerPoint slide that I'm going to send you. So I'm going to send you this PowerPoint slide with me on it, but you're going to change it to have you on it, okay? So I don't want me on it. I'm not going to put a poster out of me. So what we're going to do with these is every year for the last few years, we've made a poster of what is being done in projects, and people really liked this. They like to see what people are doing with independent projects. People within the building know about independent projects within the department, even within other departments in the building, and they are interested in what you guys are doing. So what we're going to do is have you guys make a PowerPoint slide that gives, you know, follow the template. Give your name, give your lab section. It's now Chem 256B, not just 256. Give the final structure or structures of what you're going to make, and then give a, you know, a little description of your synthesis, and then put your picture on it, okay? And you don't have to put your picture, but usually if someone chooses to not put their picture, it's like 1 or 2 people choose not to put their picture. So usually everybody wants to put their picture. Just remember, it's going to be put on a poster that's going to be displayed out in the hallway where people can see it. So kind of, you know, use discretion with the picture that you're going to choose. But make sure it is a picture that can be displayed. But otherwise, people really like to see, you know, associate your face with what you're doing, and they're really interested in what you're doing in the project. So make sure that you complete this. You'll send me this file, and then you'll also make copies of your PowerPoint slide, because the other thing we do is we kind of mark your territory in the lab by posting your PowerPoint slide above where you're going to work, okay? So then we know where your station is during independent projects, all right? Okay, let me see. I think that's, if there's anything else I need to cover, that was about it. Make sure I got all my pieces here. Oh, no, sorry. I lied. Okay, one more thing as far as the order form. The purple sheet that I gave you, that is important because those are things that we are going to have on hand for you, okay? And then the things we've had on hand all along. So the red can solvents, they're still going to be available for you. We have dry solvents. We have a dry solvent station. These are the solvents that are available as dry solvents. The acids and bases that have been available in lab will have mag sulfate. We've had mag sulfate available for you all along. It'll still be available. A lot of times you also need to use sodium sulfate, so that'll be available as well. And then also, a lot of you will have to run columns, so we'll have silica gel available too, okay? So in preparing your chem order form, I don't need to know about these. I don't need these on that order form. unless you're going to use like buckets, you know, if you're going to use like liters and liters of something, which hopefully you're not anyway, but if you are going to use like a huge amount, then include that on the order form. But otherwise, I don't need these on the order form, okay? So the reagents that you need besides these, if they're, you know, reasonable quantities, okay? Then the other thing is that green sheet. One side of the green sheet has, there's some reactions that we're going to run that are going to need either anhydrous conditions or inert conditions, okay? So they either need to be kept really dry, like our Grignard reactions were in first semester, or you need to keep out oxygen and other things out of the reaction, so you need inert conditions. So here's some lists of things that need that. A lot of times what we'll do to help these is anhydrous, definitely like Grignard, will dry your glassware, make sure everything's dry, make sure you have dry solvents, make sure your reagents are dry. So if it talks about using anhydrous conditions, pay attention to that and make sure you list things properly as far as what you need, that they are actually dry things, okay? And then keep in mind you'll have to prepare ahead of time to have things drying in the oven before you do that reaction, things like that. For inert conditions, usually what we're going to use is a nitrogen bubbler. So we have them prepared, something you haven't used before, but we'll have that available for you. Or we may do something like use a nitrogen-filled balloon over your reaction, something like that, okay? A lot of times with the dry reactions, we'll also stream nitrogen over it to keep it dry as well, okay? So that's one side. The other side is the be really careful with. Your instructor's going to have to handle these, or do not do. If you see these, we can't do these in lab, okay? So the top part is things we really need to be handled with caution, and so your instructor most likely is going to have to handle these. So if that is the case that you need to use one of these, talk to your instructor about it, and definitely make sure when you go to use this that you have coordinated with your instructor that they are there, right there, ready to help you use it. So not just you're going to set up the reaction and they're all the way on the other side of the lab helping someone else. Like, your instructor is right there helping you, okay? And this is not by any means a complete list. This is just some of the things that need to be considered. Then there's the we are not going to use these reagents list, okay? And so if anything falls in this list, we're just probably not going to use it at all unless there's a special circumstance that it will be used. Along with this, things like really high pressure reactions, we don't, besides a par shaker, we don't really have a capacity for something that's super high pressure. So par shakers usually can go 30, 40, maybe 50 psi, much greater than that, and that's of hydrogen. So anything else, any other gas that you would need really high pressure, probably going to be pretty limited by. Otherwise, really specialized equipment that we don't have access to will maybe limit what we are able to do, okay? So keep in mind as you are looking at your precedents, keep in mind the green sheet, what either reaction conditions mean, or if it's something that maybe you potentially won't be able to use because of the conditions or the reagent itself, okay? So before you leave, make sure everybody has put their names on that email list, because I will put that list together tonight, and at some point tonight, maybe later tonight, but at some point tonight, I am going to send you the blank chemical order form, the PowerPoint slide template, the inventory that's an Excel spreadsheet for the lower stockroom, and then the login information for the online portion of the inventory system, okay? Questions you guys have? Okay, here's the list. So is everybody on the list? Everyone has made it onto this guy? Yeah, I'll get